Today we are going to talk about the plant cell. Here are the various organelles of the plant cell: the cell wall, the cell membrane, the Golgi apparatus, the chloroplast, the vacuole membrane, the rapid crystal, the drusa crystal, the mitochondria, the cytoplasm, the amyloplast, that is the starch grain, central vacuole, rough and smooth endoplasmic reticulum, nucleus, nucleolus, ribosomes. What is a cell? It is the unit of life. Cell is defined as structural and the functional unit of life. Robert Hooke in the year 1665 is credited with the discovery of the cell. Some organisms such as the bacteria are unicellular. Other organisms like human beings are multicellular, that is, they are made up of trillions of cells. Now let's study about the contributions of some of the leading scientists. Swanson is called the father of modern biology. Anton van Leeuwenhoek was the first person to observe free cells like bacteria, protozoa, red blood cells and sperms. Alfonso Corti in the year 1772 observed living substances in the cells. Nucleus was first observed by Robert Brown within the cells of orchid roots. Jones Perkins J. in 1839 called the jelly-like substances protoplasm. Hugo von Moll in 1846 suggested the name protoplasm for similar substances found in the plants. Protoplasm is the physical basis of life, as stated by Huxley. Now, what is the cell theory? It was proposed by Sheldon and Chavon in the year 1838-39. It states that the cell is the structural and functional unit of living organisms. All organisms are composed of one or more cells. Rudolf Virchow said that the cells divide and the new cells arise from the pre-existing cells. Nageli in 1846 and Rudolf Virchow in 1855 modified the hypothesis of Sheldon and Chavon to give the cell theory a final shape. However, it was Louis Pasteur in the year 1862 whose experiments successfully established that life originates from the pre-existing life. Now let's answer this question. It was asked in the Ames Entrance Examination 2007. Omnis cellulae cellula was given by Virchow, Hooke, Leeuwenhoek or Brown, and the correct answer is Rudolf Virchow. Let's study the structure of an eukaryotic cell. Cell wall. It is rigid, supportive and protective covering found in plants, fungi, prokaryotes and some other protists. It lies outside the plasmalemma. The chemical composition of a cell wall is the water content is 30 to 60%, hemicellulose 5 to 15%, pectic substances 2 to 8%, lipids 0.5 to 3%, proteins 15%, microfibrils 20 to 40%, and depositions 0 to 0.24%. Parts of the cell wall. It has matrix, which consists of the following substances: water, it forms the dispersion medium of the matrix; pectin forms the colloidal complex, determines the hydration of the cell wall; hemicelluloses binds microfibrils to the matrix; glycoproteins control the orientation of the microfibrils; lipids and proteins are also found in the matrix. Microfibrils. They are the structural elements of the cell wall, formed by cellulose, and in fungi they are made up of chitin. Depositions are in the form of silica, iron and calcium, cutin, outer epidermal wall of land plants, suberin, cork cells and endodermal cells, lignin. Lignification occurs in the secondary wall of land plants. Lignin provides hardness to the wall. Now let's study about the layers of the cell wall. There are 3 layers. The middle lamella is the outermost layer of the wall, first structure that is formed between the newly formed daughter cells at the time of cytokinesis, chiefly made up of calcium pectate and some amount of magnesium pectate. The primary wall is the first laid wall which is laid inner to the middle lamella. Growth of primary wall occurs due to the incorporation of materials within the existing wall. Such mode of growth is called intersusception. Let's study about the secondary wall. It is the additional wall which is laid inner to the primary wall in some of the plant cells. The growth in secondary wall is called accretion. Pits are unthickened areas in the secondary walls of the plant cell. It is plasmodesmata. These are cytoplasmic or protoplasmic bridges between the adjacent plant cells. Plasmodesmata consists of a fine pore in the cell wall and middle lamella of 2 adjacent cells, which is lined by plasma membrane and contains a fine tubule called desmotubule. It connects the endoplasmic reticulum of 2 cells. Here we can see the structure of an animal cell and a plant cell. We find that most of the structures are similar excepting the chloroplast in the plant cell, a cell wall, functions of cell wall. The cell wall functions as apoplast. Plasmodesmata produces a protoplasmic continuum called symplast. Cell wall provides mechanical support. It counteracts the turgor pressure. What is plasma membrane? It is also known as the cell membrane, made up of 2 layers of phospholipids. They are found in all cells and contains proteins and lipids, serve as an attachment point for the cell wall. Plasma membrane is asymmetrical because the membrane-associated lipids are asymmetrical with polar and non-polar ends. They are also called amphipathic, that is, they contain both hydrophilic and hydrophobic regions. Lipids and proteins on the outer and inner surface of the membrane are different. Let's study the structure of the plasma membrane. It has carbohydrates, cholesterol, proteins, peripheral and the integral protein, filaments of the cytoskeleton, the glycolipid, the extracellular fluid and the cytoplasm. What is the chemical composition of the eukaryotic plasma membrane? Lipids are about 20 to 79% and they are identified as lecithin, cephalin and cholesterol. Carbohydrates comprises of only 1 to 5%. They are hexose, hexosamine, fructose, sialic acid, glycoproteins and glycolipids. Proteins comprises of 20 to 70% and water is 20% of its total weight. Let's see what does the sandwich model or the trilamellar model say. Daniele and Davison proposed this trilamellar model. According to this, the plasma membrane is formed of a bimolecular layer of phospholipids which is about 35 angstroms thick, sandwiched between 2 layers of proteins, each 20 angstroms thick. Thus, the total thickness of plasma membrane is around 75 angstroms. The model was proposed even before the plasma membrane was seen under the electron microscope. What is the unit membrane concept? J.D. Robertson proposed the unit membrane concept. According to this, all biological membranes share the same basic structure. They are about 75 angstroms thick, have a characteristic trilamellar appearance when viewed under electron microscope. The three layers are a result of the same arrangement of proteins and lipids as proposed by Danieli and Davison. The fluid mosaic model proposed by Singer and Nicholson. This is considered to be the latest and most widely accepted model. According to this model, the cell membrane consists of a highly viscous fluid matrix of two layers of phospholipid molecules. These serve as relatively impermeable barrier to the passage of most water-soluble molecules. The protein molecules or their complexes occur in the membrane but are not in a continuous layer. They occur as separate carriers asymmetrically arranged in a mosaic pattern. Some of these, that is the peripheral or the extrinsic proteins, are loosely bound at the protein surfaces of lipid layers. Spectrin is an extrinsic protein towards the systolic phase. According to mosaic model, the correct sequence of plasma lemma is P, L, L and P. Others are called integral or intrinsic proteins. They are in the form of single helix, example glycoproteins. Here we see the fluid mosaic model as given by Singer and Nicholson. This has the integral glycoproteins, the peripheral and the integral proteins, the extracellular space, the lipid bilayer. Fluidity of membrane. Singer and Nicholson have described cell membranes to be protein icebergs in a sea of lipids. That is, the protein component is not fixed, but it is able to shift laterally due to the fluid structure of the lipid part of the membrane. This has been confirmed by a simple but ingenious experiment devised by Fry and Eden. There are two types of wall-less animal cells. Example, mouse and human cells had been taken. One is labeled with green fluorescent antibody or difluorescin and the other with red fluorescent antibody or dirhodamine. Both are observed under the microscope to have the fluorescent pigment absorbed by the surface protein molecules of the cell membranes. The two types of animal cells are made to fuse by means of polyethylene glycol. The cells are found to have half green and half red surfaces. They are divided in two lots. One lot is kept at 0 degrees while the other is kept at 37 degrees. In the lot which is kept at 37 degrees, there is thorough mixing of the two pigments. No mixing occurs in the fused cells kept at 0 degrees centigrade, where most of the lipid tend to solidify. If these cells are kept at 37 degrees centigrade, pigment mixing occurs, showing that membrane or lipid fluidity has been restored. This is the cell fusion experiment which reveals the membrane fluidity. Functions of the plasma membrane. Cells membranes separate the cells from their external environment and cell organelles from cytosol. Cell membrane helps in cell recognition and cell adhesion. Cell membrane contains antigens which determines blood grouping. Membrane is responsible for transportation. The membrane transport are of two types in the plasma membrane. The passive transport is by diffusion or osmosis. Active transport is by sodium-potassium exchange pump or in the form of bulk transport. Again, there is a problem asked in the AIPMT 2010. Which one of the following structures between two adjacent cells is an effective transport pathway? Plasmodesmata, plasdoquinones, endoplasmic reticulum, plasma lemma, and the correct answer is plasmodesmata. The plasma membrane consists mainly of phospholipids embedded in a protein bilayer, proteins embedded in a phospholipid bilayer, proteins embedded in a polymer of glucose molecules, proteins embedded in a carbohydrate bilayer. And the correct answer is proteins embedded in a phospholipid bilayer. Endocytosis. The process of endocytosis occurs by an infolding or extension of the plasma membrane to form a vacuole or a vesicle. So, it is the process of engulfment of food substances having the size bigger than the pure size in the plasma membrane. It is of two types: phagocytosis, that is cell eating. The food material to be engulfed is taken as a solid form. The cells involved in phagocytosis are called phagocytes or phagocytic vesicles. The vesicles formed is called phagocytic vacuole. Pinocytosis is the cell drinking. Material is taken up in liquid in the form of solution, colloid or suspension. If the vesicle formed is extremely small, the process is known as micropinocytosis and the vesicle is micropinocytotic. Exocytosis or the reverse endocytosis, by which materials are removed from the cells, including reverse pinocytosis. Here we can see food particle taken in by endocytosis and it is digested, and exocytosis of waste, that is the products of digestion, are exocytosed. Cytoskeleton. The ability of the eukaryotic cells to adopt a variety of shapes and to carry out directed movements depends on the cytoskeleton. The three principal types of protein filaments are microfilaments, microtubules, and intermediate filaments. The microfilaments are made up of actin-like proteins and are capable of contraction. These microtubules are made up of alpha and beta tubulin. They do not contract but can assemble and disassemble. They play a vital role in the movement of chromosomes. In the cells where the microtubules are disrupted with chemicals like colchicine, no movement of chromosomes occur. Intermediate filaments generally form a basket around the nucleus and are present in cell-to-cell junctions. The prokaryotes do not have a membrane-bound, well-defined nucleus. Their genetic material, that is the DNA, is neither complexed with other molecules, with the histone proteins that is, and nor it is packed in the chromosome. The DNA is extensively looped and coiled with the help of nucleoid proteins called as polyamines. These proteins are different from histones present in the eukaryotes.